Welcome back to botany class. In this class, let's pay attention towards transgenic plants or genetically modified crops. So today's topic is transgenic plants or genetically modified crops. What are the learning objectives? You pay attention towards herbicide tolerant variety and insect resistant varieties. There are two types of question you are going to learn. Under transgenic plants, there are many different types of transgenic plants. What are transgenic plants? Modified, genetically modified plants. Many varieties are there, but you are going to concentrate only to herbicide tolerant variety and insect resistant variety. Herbicide tolerant, insect resistant, and its advantage and disadvantage. Let's go into the class. Herbicide tolerant. Before that, what's called transgenic plants? You know that in this chapter you are continuously learning about gene modification, how the plants or the tissues or the organisms or microbes can be modified by introducing the desirable genes. Such a way, these transgenic plants are genetically modified crops. The crops can be modified by introducing certain types of genes. Such a way, the genes or the plants are modified and create a new variety called transgenic plants. Mainly used in plant breeding technology for herbicide tolerant and insect resistant. What are these herbicide tolerant? So transgenic plants which contains a novel DNA. Novel DNA is the unusual DNA which can be incorporated inside the cell, modified form. This herbicide tolerant, you know that in the crop plants, these weeds, herbicide, herbs, unwanted plants that are called weeds. These weeds reduce the yield, crop yield, which reduces. So normally the farmers, they use herbicide. The herbicides are the chemical substance which used to kill or eradicate the unwanted herbs that are called as weeds. To remove or eradicate weeds, farmers or the plant breeders, they are applying herbicides. So when they are applying herbicides, these herbicides not only affect the herbs, that is unwanted plant like weed, it not only affect the weed, it also affect the crop plants. To avoid this, they are making this plant as a transgenic. So it has to tolerate herbicide tolerant variety. If the plants are modified in such a way which can be able to tolerate the herbicide, because the herbicides which contains the chemical substances which is toxic to the food items in the form of the crop yield. So here there are two different types of herbicides. You are going to learn there are two different types of herbicide. So these herbicides, the weeds are creating main problem to reduce the crop yield. So these weeds are compete not only against the sunlight, then food or nutrient or water, which also the carriers of disease and insects. So weeds already which compete towards this, against this, compete against the crop field, crop plant. Not only for sunlight, they are competing among themselves for sunlight, water as well as nutrient. Not only this, it also compete for carrying insects as well as diseases. So if it is uncontrolled, this create main problem to reduce the crop yield. That's the reason It's important to make this plant as a herbicide tolerant variety. There are two different types. So here there are two different types of herbicides. They are glyphosate and basta. Glyphosate, number one, there is a glyphosate. Glyphosate is a herbicide. So this herbicide is made from the company Monsanto from USA. The trade name of this is Roundup. So this glyphosate herbicide, which contains an enzyme that's called 5-enopyruvate shikimate 3 phosphate synthase enzyme. The enzyme which encodes in this herbicide is named as 5-enopyruvate shikimate 3 phosphate synthase enzyme. So it's commonly called as this first letter of this one, ESPS enzyme, ESPS, EPSPS. So glyphosate is the herbicide which made from the company Monsanto from USA. The trade name of this is Roundup, and it encodes an enzyme EPSPS called as 5-enopyruvate shikimate 3 phosphate synthase enzyme. So this enzyme inhibits the process of vital the amino acid vital nutrients secretions. So this process can be affected by this enzyme. It inhibits the process of this amino acid synthesis. So there are some strategy to make the transgenic plants against this enzyme EPSPS. So there are several methods to introduce or incorporate the plant cells, but the two methods are, one of the strategy is by incorporating the cells. The genes are incorporated to the cells by produce the tolerant of this EPSPS. The enzyme is enopyruvate shikimate 3 phosphate synthase enzyme. To produce, when this genes are incorporated into the plant cell, the cell has the ability to tolerate against the activity of this enzyme EPSPS. One strategy. Another strategy is, so this bacteria, this gene can be incorporated by the soil bacteria. Bacteriums are used because vector is needed. It's selected from the bacteria. Another strategy is, one is to produce against the to tolerate this. Another one, degrade the activity of this enzyme EPSPS. So first question, there is herbicide tolerant. Listen carefully. Herbicide tolerant glyphosate, herbicide tolerant variety. This plant, now the topic is transgenic plant. What are transgenic plant? Genetically modified crops. The crops can be modified in such a way to tolerate the herbicide. Here what's the herbicide? Glyphosate. To tolerate glyphosate, there are two strategies are used for incorporating the gene. They are using soil bacterium. In this, the first strategy to introduce the gene from the bacteria to produce the tolerant of this enzyme EPSPS. Another one, to degrade the enzyme activity. So this enzyme which produces amino acids, to degrade the activity of this enzyme and made this plant as a transgenic. Another example is Basta. Basta is another herbicide. So this herbicide Basta is a non-selectable herbicide. It's a non-selective herbicide. Non-selective. Non-selective, it do not have any specific host. Commonly it attacks the herbicides. So non-selective herbicide which contains a chemical compound. All the herbicides which are made up of chemical substances. So these chemical substances, herbicides are chemical substances which not only affect the herbicides alone, herbs alone or the weeds alone, unwanted herbs or the weeds alone, which affect the crops also. So this contains a chemical compound, Basta, which contains a chemical compound, that is phosphinothrycin. Phosphinothrycin. Here you are going to learn so many chemical names, enzymes. The names are very important for 1 word, 2 mark, as well as the 3 mark, the activities of this. You will learn all this with correct spellings. Chemical compound phosphinothrycin. Phosphinothrycin is a chemical compound present in Basta. What is Basta? Basta is a herbicide which contains a chemical compound phosphinothrycin. Phosphinothrycin is the chemical compound. So here this plant is modified by herbicide tolerant gene. There is a herbicide tolerant gene that is called PPT. Here in herbicide tolerant gene for Basta is PPT, which is known as L-phosphinothrycin. L-phosphinothrycin, PPT. So phosphinothrycin is herbicide tolerant gene. The chemical compound present in Basta is phosphinothrycin. The herbicide tolerant gene is phosphinothrycin. So PPT, L-phosphinothrycin. So this has to tolerate against. When you are modifying the plant, it can tolerate this chemical compound phosphinothrycin. So they are selecting a gene PPT. This gene can be isolated from a plant. The gene is isolated from a plant named as Medicago sativa. Name of the plant is Medicago sativa. Medicago sativa. Medicago sativa. From that plant, the gene is isolated, and this gene inhibits the activity of glutamine synthase. There is another enzyme called glutamine synthase. So this glutamine synthase, this activity of this glutamine synthase, that is the process of this enzyme is ammonium assimilation. It affects or inhibit ammonium assimilation. So this PPT gene, the herbicide tolerant gene PPT, can be introduced into tobacco plant. This gene is introduced into tobacco. So tobacco plant is making as a transgenic against the herbicide Basta. So the gene, herbicide tolerant gene is PPT. Similarly, the same gene, it is isolated from Streptomyces hygroscopicus. Streptomyces hygroscopicus. So this Streptomyces hygroscopicus contains a bar gene which encodes a gene called bar gene, that is PAT. That means phosphinothrycin acetyl transferase. Phosphinothrycin acetyl transferase. Phosphinothrycin acetyl transferase, PAT. That is PPT. Example tobacco. This is PAT, the bar gene from Streptomyces hygroscopicus. Streptomyces hygroscopicus. From Streptomyces hygroscopicus, the gene encodes bar gene it encodes the gene PAT, and this PAT is introduced into potato and sugar beet plant. So here two examples you studied. One is tobacco, transgenic plant, herbicide, basta. Transgenic herbicide plant is tobacco. Tobacco, the introducing gene is PPT, which is isolated from which is introduced into tobacco plant, isolated from Medicago sativa plant. Medicago sativa. But here Streptomyces hygroscopicus PAT. This is PPT, here PAT. Phosphinothrycin acetyl transferase. So two examples under herbicide resistant basta. Basta, you are going to write two examples. One is tobacco, modifying tobacco plant. Another one is modifying potato and sugar beet. Potato and sugar beet, the enzyme, the gene is isolated from Streptomyces hygroscopicus. The name of the gene is PAT. And tobacco modified plants, the gene is isolated from Medicago sativa. The name of the gene is PPT. Now, what are the advantages of this herbicide varieties? What's the advantage? Is there any advantage to this transgenic plants? Why they are modifying this plants? If there are any advantage, they can modify. What are the advantages? So, weed control. So, when you are applying this transgenic plant, when the plants are made as a transgenic, which has the ability to control the weed, automatically when the weeds are controlled, the crop yield can be raised. So, high yield. The weeds which reduce the crop yield, if you avoid the weeds by making the plants as a transgenic, which produces high yield. So, this is one of the advantage. Crop yield can be higher crop yield, and it reduces the spray of herbicides. Normally, we know that paddy field or any other field, any kind of field, the herbicides can be sprayed. So, here the spraying amount of herbicide can be controlled or it can be reduced. No need of spraying herbicides because the plants can be tolerated. Plants can be modified, transgenic, which can be modified or transgenic to tolerate the herbicides. So, which cannot be affected. Then, it reduces the competition between weeds as well as the crop plant. So, these plants, these weeds automatically which grow along with the crop plants. There will be a competition among these two, the weed as well as the crop plants. So, this competition can be avoided when the plants are transgenic. Then, use very low toxicity compounds. So, it should avoid the spraying of herbicide. So, herbicide which contains chemical compounds that is toxic to the crop yield. Now, when the plants are transgenic, it cannot be toxic. The amount of toxicity can be reduced, and also it conserves the soil and the soil microbes. There are number of microbes present in the soil. That microbes can be ability to conserve. We are conserving the soil as well as the microbes present in the soil. Or else when we are spraying this insecticide, the chemical compounds which are placed in the soil, located in the soil, and it polluted the soil and it affects the fertility of the soil. Now, it's one of the advantage to conserve the soil and the soil microbes. There are number of microbes which are helpful to the farmers or which increases the crops, like earthworm and some other soil worms, soil organisms, microbes. So, these microbes can be conserved. So, these are the advantages. So, main advantage is it improves the yield. It makes the crop high yield. It gives high yield when we are making the plant as a transgenic. Then another one, so first one, transgenic herbicides. So transgenic plants, there are so many: disease resistant, pest resistant, insect resistant, herbicide resistant. Herbicide resistant, the first question, today's first question, you learn about herbicide tolerant. Herbicide tolerant. Example, two examples you study: tolerant of glyphosate and tolerant of Basta. Now second question is insect resistant varieties. First one, herbicide tolerant variety. Now insect resistance varieties. Another important problem in the crop field is insects. Insects like pests, worms, butterflies, somas, so many insect beetles which affect the crop. So it not only affects the crop, it affects as well as it spreading diseases sometimes, some kinds of insect which spreads diseases. So insect resistant varieties. So these plants are modified as a genetically GM crops, genetically modified plants or genetically modified organisms. Here BT crops, BT, Bacillus thuringiensis. The main bacteria which is used for insect resistance variety is Bacillus thuringiensis. So there are three examples you are going to learn. Number one, BT cotton. There are three examples. One is BT cotton, BT brinjal and Dara mustard hybrid. So here three plants: cotton, brinjal and mustard plants. Cotton, brinjal and mustard. So first example is BT cotton. BT cotton. That is GMO. What is meant by GMO? Genetically modified organisms. Here the organisms can be modified as a insect resistant. So second question is insect resistant varieties. So in this BT cotton, the insect resistant varieties, the insecticidal activity against bollworm. In cotton plants, there is a worm called bollworm which comes and eat the cotton plant. So insecticidal activity is bollworm. So here the bacteria which is used to transgene is Bacillus thuringiensis, BT. That's why it's called as BT, Bacillus thuringiensis is the bacteria. You may learn in the lower classes also. Bacillus thuringiensis is used for insect resistant varieties. Bacillus thuringiensis, which contains, there are over 200 BT toxins, or this all 200 BT toxins are available in Bacillus thuringiensis. The Bacillus thuringiensis bacteria which encodes endotoxin, this enzymes. So toxic substances are secreted in its body. So over 200 BT toxins are present which are harmful to insects. That's why they are selecting this Bacillus thuringiensis. This all enzymes, there are over 200 enzymes can be isolated as a toxic substance against the insects. So these are harmful to insects. These all toxic substances are toxic crystals. They are the toxic crystals. So they are the endotoxin crystals. They are coming under the group CRY groups, CRY groups. These substances, toxic substances, are dissolved in the insect stomach. When the bollworms come and eat the plant parts of the cotton, what happens? This toxic substance, endotoxin, dissolves in the insect stomach. So the epithelial membrane of its gut, which blocks the activity of regulation of potassium ions. So there are number of activities taking place in the epithelial membrane. When it is dissolved, when these toxic substances are dissolved in the gut of this insect, It affects the normal function. It affects to take the vital nutrients. And one among this is it blocks the regulation of potassium ions. So the function of this enzyme is it affects or it blocks the regulation of potassium ions in the guts. So what happens? This epithelial cells which started to degrade, due to this it causes death of the larva. The larva die because of this toxic substances. It degrades the epithelial membrane which is present in the gut or belly of this insect. So because of this endotoxin which affects this activity, potassium ion, regulation of potassium ions. This potassium ion regulation blocks by endotoxin. The endotoxin belongs to the group Cry group, that is toxic crystals, Cry groups. So Cry group endotoxin affects the regulation of potassium ions. It leads to death of the larva. This question, Bt cotton, genetically modified organism. One of the genetically modified organism is Bt cotton. The insecticidal activity against bollworm. You remember this cotton plant, there is a worm, bollworm. The name of the bacteria is Bacillus thuringiensis. There are over 200 toxic substances can be derived from this Bacillus thuringiensis. These toxic substances are harmful only to the insects. Insects which includes butterflies, moth or larva, this bollworms, these all types of insects. And this insect, the bollworm, when come and eat the plants of this cotton, this toxic substances, it affects the stomach of this insect. It dissolves. The toxic substance dissolves the stomach and it degrades the epithelial membrane of gut. It leads to death of the organism, larva, because of blocking the regulation of this potassium ions. So what's the advantage of this Bt cotton plant? What are the advantages of Bt cotton plant? So yield can be increased. This all transgenic plants which improve it increases the yield. So here also in the cotton plant, when we are making this as a GMO, genetically modified organism, automatically the yield can be increased. The cotton yield increased. And reduction of usage of insecticide. So when the plants are transgenic, when the plants are modified as transgenic, no need of using insecticides. So that is another advantage. And reduction of this cost of cultivation also. So cost of cultivation, otherwise we can make some amount to spraying the insecticides, pesticides. Each and every insect can be used. But when the plants are tall, which is transgenic, no need of using extra amounts. Then what are the disadvantages? So genetically modified plants can advantage. Also there are some disadvantages. What are the disadvantages? The cost of this seed. Here what seed? The cost of the Bt cotton seed. Now this seed is Bt cotton seed. So it is transgenic seed. Normal seed we cannot plant it. Transgenic plants, it is somewhat cost than the normal. And effectiveness too, the effectiveness of this plants up to 120 days. So effective of this seeds is up to 120 days. And this effectiveness after 120 days, it can be, the effect of this plant can be reduced. So this is one of the disadvantage. And this ineffective against other insects, like for example, there are some other insects like sucking pest, some kinds of pests are there. These enzymes are, this delta toxin is not harmful to such pests like aphids and white flies. It's not harm. So it is ineffective against sucking pest. This is effective only to this bollworm. Other insects is not effective. And it affects pollinating insects. So it reduces the yield also. So sometimes it affects the pollinating insects. So many insects or visit the plant to as a visitors, agents of pollination, pollinating agents. It will be affected. So these are the disadvantages. Now let's see another example, brinjal. BT brinjal. This is the example, BT cotton. Next question is BT brinjal. So here BT brinjal is a brinjal plant. So in this modified transgenic brinjal, here there is a protein gene insertion of how to make this transgenic, the brinjal plant, inserting a crystal protein gene. Here another gene, insertion of another gene, crystal protein gene, which is under the Cry group CRY1AC. The name of the gene is CRY1AC. This is inserting. This is also isolated from Bacillus thuringiensis. This BT brinjal and BT cotton, which is isolated or derived from Bacillus thuringiensis. And this gene, here what is the gene for brinjal? Crystal protein gene called CRY1AC. CRY1AC. So this crystal protein gene insert, which is isolated from Bacillus thuringiensis, insertion along with genetic elements. So when we are inserting this gene into a organism, there is a vector used. Here the vector is used as Agrobacterium tumefaciens. So all kinds of plants, normally we are using Agrobacterium tumefaciens. So along with this insertion of genetic elements. What are the genetic elements? If the vectors, when we are introducing a vector, the vector contains a promoter, terminator and antibiotic resistant marker. So this is called genetic elements. So insertion along with genetic elements. Genetic elements including promoter. The vector which contains a promoter. Promoter is a region which initiate the transcription process. It's a sequence of DNA which initiate the process. Terminators are the end region of the DNA and the antibiotic resistant marker gene. Marker gene also there. So along with this, the insertion using Agrobacterium tumefaciens. The gene is inserted. So which is resist against the insect, the variety of insect Lepidopteran. Lepidopteran insects. So Lepidopteran insects includes butterflies and moth. So this is tolerant. It resists against this kind of insects called Lepidopteran, like butterflies and moth. And there are some other insects which visits this brinjal, fruit and shoot borer. The fruit and shoot borer, some insects which with their proboscis it bores the stem part or the fruits, the shoot or the fruit. They are called as Leucinodes arbonalis. Leucinodes arbonalis. Leucinodes arbonalis is the kind of insect, fruit and shoot borer insect. So under this BT brinjal, insertion of a gene, the name of the gene is CRY1AC, that is crystal protein gene, isolated from Bacillus thuringiensis and used, insert it along with genetic elements with the help of Agrobacterium, using the bacteria, soil bacterium, Agrobacterium tumefaciens, which is resistant against Lepidoptera larva. So, Lepidoptera insects, butterflies and moths. Then another question, Dara mustard hybrid, mustard. So, here you learned resistant, disease resistant varieties, insect resistance. Insect resistant varieties includes cotton, then brinjal and mustard. The name of the mustard is Dara mustard hybrid, which is called as DMH. That means Dara mustard hybrid, DMH. The name of this plant is DMH11. So, this mustard is a transgenic mustard, is developed by a team of scientists from the Center for Genetic Manipulation for Crop Plants. Genetic manipulation of, they are from the Center of Genetic Manipulation of Crop Plants from Delhi University. Is under a government project, a government sponsored project. They produce this DMH, Dara mustard hybrid plant. So, this is herbicide tolerant variety. So, this variety is herbicide tolerant variety, and this tolerant mustard using the enzyme, that is Burnase-Burstar enzyme. Burnase-Burstar technology, using a technology here, Burnase-Burstar technology. This technology, they are creating a seed, hybrid seed, making a hybrid seed by male sterility. So, it is a technology, they are making a hybrid seed, Burnase-Burstar technology. So, they are adding a gene from the soil bacterium. They selected the soil bacterium and add the gene and make this plant as self-pollinating plant. Self-pollinating plants which encodes 3 genes. This bursa, using this Burnase-Burstar, which contains 3 different genes. They are Bar gene, Burnase gene and Burstar gene. 3 different kinds of genes present in it. They are Bar gene, Burnase. The name is Burnase-Burstar, which contains Bar gene, Burnase gene and Burstar gene. So, here this Bar gene, which is resistant to tolerant against Basta, you remember this bar, tolerant against Basta. So, this is the name of the plant is DMH11 mustard plant, Dara mustard hybrid plant. So, insect resistant, you learned 3 different examples, BT cotton, BT brinjal and mustard, DMH11 mustard. Once again, look at this, what are the questions you are going to learn today. Transgenic plants, there are 2 different types, one is herbicide tolerant, example glyphosate and Basta. Then insect resistant, there are 3 different plants, that is BT cotton, BT brinjal and Dara mustard hybrid variety. Learn all these questions and learn all the spellings correctly. Prepare well for the exam. Thank you.